So hello guys, welcome back to my YouTube channel, that is Learning Biophysics with Team 320, and this is the week 6, and the today is my topic is types of muscles and their structure and the types of muscle proteins. So first of all, I will start with the muscles. Muscles is defined as the tissue primarily composed of specialized cells, fibers which are capable of contracting in order to effect movement, like we are this movement or any of the movement in the body is because of the muscles. This can relate to the movement of body or a body parts with our external environment. And however, did you know that almost all movement of the blood, food and other substance within the body is also due to muscle contraction? Depending on the type of the primary function of the muscle is to move the bones of the skeletal. However, muscle also enables the heart to beat and can be found in the walls of hollow organs such as you can see in intestine or uterus, stomach, everywhere. For like hollow, these muscles are have a primary functions. So in this topic we will see the types of muscles, that is striated muscle, skeletal, visceral, striated or cardiac muscles, or non-striated muscles, that are smooth muscles, and skeletal muscles. It's just an overview. I will talk it about in deeply. So skeletal muscle, striated muscle formed of long multinucleate unbranched myocytes attached at one or either ends of to the bony attachment point. This is just an overview for the skeletal muscle. Similarly, with as a cardiac muscle is a striated muscle formed of short uninucleate branching myocytes which connected at intercalated disc, and a specialized muscle of the heart, that is myocardium. Next muscle is the smooth muscles, non-striated muscles formed of short uninucleate spindle-shaped myocytes located in the walls of the internal organs and blood vessels, etc. Connective tissue, and when we see the connective tissue over here, that is endomysium, perimysium and epimysium. Endomysium, that is loose connective tissue surrounding muscles, cells, fibers, and perimysium, that is fibrous sheath which divides muscles tissue into fascicles, and epimysium, that is fibrous sheath which surrounds entire skeletal muscles. And further we see overview of the muscles, that is muscle has four main properties, that is excitability, contractibility, extensibility and elasticity. And so excitability is an ability to respond to stimuli, contractibility and ability to contract, extensibility and ability to be stretched without tearing, elasticity and ability to return to its normal shape. Through these properties, the muscular system as a whole performs several important functions. These include the production of force and movement, support of the body structure, position, stability of joints, for production of body heat to maintain normal body temperature, as well as provision of form to the body. So when we see the structure of the muscles, regardless of its morphology or type, muscles tissue is composed of specialized cells known as muscle cells or myocytes. From the Greek word, we can say that myo means it is like originated from the Greek word mys, that means muscle. So commonly referred to as a muscle fibers. Myocytes are characterized by protein filaments known as actin and myosin that slides past one another, producing contraction that move body parts, including internal joints. Interestingly, these proteins are not exclusive to muscle cells. Actin myosin are also commonly known as cytoskeletal element, or we can say the in many cell types and are involved in cellular functions relating to the changing of the cell shape. Example, we can say cell movement, phagocytosis, etc. Myocytes, however, are characterized by a particular abundance of this protein within their cytoplasm, so much as that they occupy most of their interior. Furthermore, in the case of Myocytes, actin and myosin filaments are generally oriented along in a single axis, thereby eliciting movement in a linear fashion. Now we can see that muscle tissue is classified as either striated or non-striated, that is smooth, striated, that is skeletal, visceral and cardiac, that I have already told. Now I will tell you about the skeletal muscle. In skeletal muscle, you can see that this diagram is shown on the screen. Skeletal muscle is the most common type of muscle tissue found in the body and consists of highly elongated, multinucleated, non-branching cells which are arranged in a parallel manner. Skeletal myocytes often measure several centimeters or 10 of centimeters. They are biologically classified as syncytia cells, which are formed by the fusion of several similar smaller mononuclear cells. In this case, in the case of skeletal muscle, the cell which merge to form monocytes are known as myoblast. Skeletal muscle is often referred to as a voluntary muscles due to the fact that we think of its contraction being under conscious control. This is a misconception, we can say. However, as a skeletal muscle is involved in various movements which are under subconscious level, that is we can see subconscious level, like skeletal muscle while breathing, we can say it's a subconscious level, and it is like voluntary. Being a voluntary muscle also, it is working as a subconscious level also. So sarcomere, if we have you seen the diagram of the this skeletal muscle, the first part we will study about the sarcomere. The cytoplasm of the each skeletal muscle fibers, myocytes, referred to as a sarcoplasm. Sarco commonly used to denote muscle related term, that is flesh, Greek word that is sarx, is largely occupied by the subunit known as myofibrils, which extend the length of the cell. Myofibrils are essentially changed structure composed of repeating unit of contractile units known as sarcomeres. These are primarily composed of the two protein myofilaments, thin actin and the thick myosin, and used for muscular contraction. So now we will see the muscle contraction. In the video I will show how muscle contraction occur.
Striated muscle is made up of bundles of multinucleated muscle cells called myofibers. A myofiber is packed with myofibrils that extend the length of the cell. A sarcomere, the functional instructional unit of muscle, consists of thick myosin filaments and thin actin filaments. Titin keeps thick filaments centered during contraction. In muscle contraction, actin filaments slide past myosin filaments. Neither filaments change in length, yet the sarcomere becomes shorter. The force required for this movement is generated by myosin heads, which make cross-bridge attachments to actin. Myosin heads walk along an actin filament, using ATP for energy. Tropomyosin is part of the complex that prevents myosin from sliding along the thin filaments. A myosin head binds ATP, and as a result, detaches from the actin filament. It rebinds in a new position. Phosphate release triggers a power stroke that moves the actin filament. ADP dissociates, and the process is repeated.
So as you seen in the this video, how muscle contraction occurs. Now we will further move to the next muscle group, that is cardiac muscle. Like skeletal muscle, cardiac muscle is striated and hence is composed of similar contractile proteins which are also structurally arranged into sarcomeres discussed above. It is however where most similarities between these muscle tissues types ends, like cardiac muscle cells, cardiomyocytes are much shorter and broader than the skeletal muscle and are branched at their ends. They are generally uninucleate, that is, have one nucleus each. However, sometimes may be binucleate. The nucleus is centrally located compared to those seen peripherally in the skeletal myocytes, with the myocytes passing on either side, having myofibrils on the either side, leaving a clear zone of perinuclear sarcoplasm around the nucleus. You can see on the diagram that is being shown on the screen, that is transverse section of cardiac muscle and longitude section of the cardiac muscle. We can see on this diagram cardiac muscle fiber, branched cardiac muscle cells, intramuscular capillaries, cardiac muscle fascicles, cardiac muscle cells, cardiac muscle cell nuclei, striations, intercalated disc, endomysium, and the last is fibroblast nucleus. So striation in the cardiac muscles are not as defined as those seen in the skeletal muscle, as they are slightly obscured by relatively large amount of mitochondria and the organelles present in the cells. Cardiomyocytes are surrounded by fine, loose connective tissue similar to endomysium seen in skeletal muscle, albeit less organized. Condensation of perimycal like dense connective tissue can be observed, dividing groups of cardiac muscle cells fibers into fascicles, which, unlike that seen in the skeletal muscle, whirl and spiral in a multi-directional manner, except in the case of papillary muscles. And as a result, cut section of cardiac muscles tissue will usually present various orientation of muscle fibers adjacent to one and another. So next we can see the diagram here, that is pacemaker cells and innervations diagram. You can see on the screen. Here is the heart, where we can say that on this diagram of the heart, you can see sinoatrial node, atrioventricular bundle of Hayes, left bundle branch and right bundle branch, sub endocardial branches of atrioventricular bundle. So this is the conduction system of the heart. Cardiac pacemaker cells. Now this is the main main cells of the cardiac tissue. So we can see that this stimulating cardiomyocytes, cardiac conducting cells, are highly specialized modified cardiomyocytes which are capable of generating and carrying contractile signals. Why I have shown that diagram? So that you can understand that how the signals are passed through with the help of these muscles and these cells. So these signals is due to cardiac pacemaker cells, conducting system of this due to pacemaker cells. These are arranged into nodes, that is sinoatrial nodes and the atrioventricular nodes, SA node and AV node we basically say, and atrioventricular sector, that is section, that is bundle of His, and sub and endocardiac conducting network, commonly referred to as a Purkinje fiber, to form the conducting system of the heart. So these cells, these muscles helps heart to conduct helps to work as a conducting system of the body. So next the next muscle we will talk about is the smooth muscles. In smooth muscle is most commonly found in the walls of tubular structure, example vessels, gut, ducts, bronchi, etc., as well as hollow organ, that is urinary bladder, uterus, and principally functions to modify the diameter size of this structure. So you can see the diagram of the longitudinal section and transverse section as we have shown in the cardiac. There is a smooth muscle now. So you can see nucleus of the smooth muscle cells, that is sarcoplasm, smooth muscle cells, fibroblast nucleus. On the second diagrams, you can see myenteric plexus of Auerbach and the smooth muscle bundle connective tissue, This is, you can see in the smooth muscles histology. Like smooth muscle cells are generally uninucleate and are much smaller and shorter than those seen in skeletal muscle. They are spindle shaped with long tapered ends and are usually packed together with their long axis parallel to the neighboring cells in an interdigiting manner. Each cell is enveloped by a basement membrane and other connective fibers which breach the spaces between adjacent cells. Condensation of these extracellular structures known as dense plaques provide the region of attachment for the smooth muscle cells. So yes, this is all about I want to show one diagram to you all on the screen. You can see this diagram like dense plaque, dense body, actin, myosin, uncontracted state and the contracted state, how this differs in the smooth muscles. So you can see this diagram, and smooth muscle like cells are also present in the smooth muscle section. We can talk about that. That is pericytes, myoid, peritubular cells, that is testis. Pericytes found in the capillary walls, myoepithelium cells often found in glandular epithelium, myofibroblast, that is skin wound healing. And this is all about the smooth muscle also.